Hello students. In this video, we'll be solving few questions about protein folding. Now, question 1. Given below are thermal melting curves for protein A, P, Q and R. The x-axis denotes temperature, y-axis indicates the concentration of the unfolded protein. Now in this curve, with increase in temperature, more and more of the protein will become unfolded. Now, which one of the following options is true regarding comparative thermostability of protein A with respect to P, Q and R? So our options are, first option is A is less stable than P, A is more stable than Q and R. Our second option is A is less stable than P, A is more stable than Q and A is equally stable to R. Our option 3 is A is similarly stable to B and A is less stable than Q and R. Our fourth option is A is similarly stable to P and A is less stable than Q, but A is more stable than R. So how will we solve this? So let's try to first focus on one of the comparisons and see if we can narrow down our answers. So let's compare A with Q. So as we can see in this graph, at the same temperature, the unfoldings of protein Q and protein A are different. We can see here that at the same temperature, the level of unfolded Q protein is much lower than that of unfolded A protein, which means that A is less stable than Q. So this narrows down our options to option 3 and 4. Now since the comparison of A and P is same in both the options, we just have to consider the third parameter, comparison of A with R. Now for comparison of A and R, we can see that they reach the same level of unfolded proteins but at different temperatures, which means that it takes a higher temperature for the protein R to become unfolded compared to protein A, which means that R is more stable than A. So A is less stable than both Q and R. So our correct answer will be option 3. Moving on, question 2. 3 strands of a beta sheet of 4 peptides are hydrogen bonded with the orientation of the strands parallel or antiparallel denoted by the arrowheads as shown in the figure. Each strand consists of identical residues, which means same amino acids, where N and C represent terminal residues of the peptide. The 3 strands of each peptide are linked by amino acid sequences of the smallest length possible. Now, which one of the following option is correct regarding the length of the peptides 1 to 4? So we have to arrange all the 4 peptides in the increasing order of the length. So our first option is peptide 1 and 2 are of equal length, peptide 3 is longer and peptide 4 is longest. Option 2 is peptide 1 is the smallest, peptide 3 and 4 are of equal length but longer than peptide 1 and peptide 2 is the longest. For option 3, the increasing order of the length is peptide 1, 3, 4 and 2. And for option 4, the peptides in increasing order of the length are peptides 1, 2, 3, and 4. So let's solve this. What we'll do is, first we'll link all the strands in one single peptide, like this, linking the C terminal to the N terminal. In all these strands, the arrowhead is the C terminal and the flat side is N terminal. So as we can see in the peptide 1, that both of the linkers are of same length. Now moving on to peptide 2. Because of the orientation, the linker that links the C terminal of the first strand to the N terminal of the second strand is longer compared to peptide 1. Now for peptide 3, the first linker is longer, but the second linker is shorter. And for peptide 4, first linker is shorter and second is longer. So what we can see from this image is that peptide 1 has two linkers, both of which are short, and peptide 2 has two linkers, both of which are long, which means that peptide 1 is the shortest and peptide 2 is the longest. Now peptide 3 and 4 both contain one linker of the longer length and one of the shorter length, making both of them equal in length. So from this, we can say that our answer will be option 2. Moving on to question 3. In a protein stability study, 3 solutions of protein mol A, which is of 10 kilodalton weight at 0.5 millimolar, mol B, which is of 20 kilodalton weight and again at 0.5 millimolar, and mol C, which is of 20 kilodalton at 1 millimolar, were subjected to denaturation by urea SDS and guanidinium hydrochloride respectively. The profiles of the fractionation of unfolded protein with increasing concentration of the denaturants is given below. Now, which one of the following corresponds to reaction conditions at which the number of molecules of folded protein are equal, assuming the reaction volumes to be same for all experiments? Now, what we can see from all these graphs is that the x-axis denotes the concentration of the denaturant and the y-axis denotes the fraction of the proteins that are unfolded. So as it is already provided in the image, for the urea at 2 molar concentration, half of the proteins are unfolded for mol A. And again, for mol B with the SDS at 0.25% SDS concentration, half of the protein molecules are unfolded. And for third protein, mol C, with the guanidinium hydrochloride as a denaturant, at 4.5 molar concentration, half of the molecules are unfolded. Now let's look at our options. Option 1 is 0.2 molar urea, 0.05% SDS, and 4.5 molar guanidinium hydrochloride. Option 2 is 2 molar urea, 0.05% SDS, and 1 molar guanidinium hydrochloride. Option 3 is 0.2 molar urea, 0.25% SDS, and 4.5 molar guanidinium hydrochloride. And option 4 is 2 molar urea, 0.25% SDS, and 1 molar guanidinium hydrochloride. Now looking at the charts for mole A and mole B, the answer is already given in the question. The concentration of the denaturant at which half of the proteins are unfolded for urea is 2, and for SDS it is 0.25. Now looking at the options, only one of the options has both of these numbers, that is 2 molar urea and 0.25% SDS, that is option 4. Now they have mentioned the molecular weights of the proteins in this question to confuse you, but since the concentrations of all the proteins are already given and the graph shows the fraction unfolded, we don't really need the molecular weights. Just from the graph we can get our answer. Now moving on to question 4. The amino acid sequence of tetrapeptides P, Q, and R is shown below. Peptide P contains aspartate, glycine, aspartate, and serine. Peptide Q contains glycine, serine, arginine, and glycine. Peptide R contains glycine, lysine, arginine, and alanine. So the first question is, calculate the net charge on the above tetrapeptides at pH 7. Question 2 is, if the mixture of the above tetrapeptides is separated on a cation exchange column at pH 7, which tetrapeptide will elute last? So we'll have to calculate the charge of the tetrapeptide. So what are the sources of positive or negative charge in the amino acids? So the sources of positive charge are free N-terminal amine group, lysine side chain, and arginine side chain. Now, sources of the negative charge are free C-terminal carboxylic acid group, aspartate side chain, and glutamate side chain. Now, with this information in mind, let's calculate the charge for first tetrapeptide, that is aspartate, glycine, aspartate, and serine. Now, when dealing with any polypeptide, the N-terminal and C-terminal groups will cancel out their charge, which are +1 and -1. So we just need to consider the charge of side chains when we are calculating at pH 7. If the pH is differing from 7, then you will have to consider the terminal groups depending on the pH. But since we are calculating for pH 7, we just need the information of the side chains. Now for the first peptide P, there are 2 aspartate groups, which means 2 negative charges, that is -2. And if you look at the options, you can see that only one of the options has the charge of P as -2. So that is our answer right there. Still, let's count the charges for all the other 2 tetrapeptides. The peptide Q is glycine, serine, arginine, and glycine. Again, the contributing group is only one, arginine side chain. So the charge for Q will be +1. And for the tetrapeptide R, which has the sequence of glycine, lysine, arginine, and alanine, there are 2 contributing groups, lysine and arginine side chains. So its charge will be +2. And since the peptide R is the most cationic, it will be the last one to be eluted when going through a cation exchange column. So our answer will be option 1. Now to deal with this type of questions, you'll need to remember 2 things. First, chemical structures of all the amino acids. and the second is the pKa values for the all of the side chains of the amino acids, because this question can be made much harder by changing the pH from 7 to any acidic or basic number. Now moving on to question 5. This will be our final question for today. So the question is, you have purified an enzyme that has a molecular weight of 60 kilodaltons. You run this protein on an SDS page gel and get the following results. So in the gel image, we can see that we are getting two different bands, one for 30 kilodalton and one for 10 kilodalton. Now, which of the following statement is valid for the quaternary structure of this enzyme? First option is 2 subunits of 30 kilodalton. Option 2 is 1 subunit of 30 and 3 subunits of 10 kilodaltons. Option 3 is 6 subunits of 10 kilodalton. And option 4 is 3 subunits of 30 kilodalton and 1 subunit of 10 kilodalton. Now, to answer this, we can see in gel that there are two different bands of 30 and 10 kilodalton, which means that our answer will contain both the subunits. So it leaves us with option 2 and option 4. So now total weight of our enzyme is 60 and the weight of the subunits is 30 and 10. Now let's consider option 2, 1 subunit of 30 and 3 subunit of 10 kilodaltons. So total will be 60 kilodalton, which is our answer. Now considering option 4, 3 subunits of 30 and 1 subunits of 10, the answer will be, the result will be 100 kilodalton, which is not the weight of our enzyme. So our answer will be option 2. If you had any trouble with any of the questions that we solved today, let me know in the comments. As usual, I'll try to explain them in a better way. Thank you for watching.